All right, folks, thank you so much for being here today. Okay, we can skip right through that. The key thing about this slide, there's a conflict of interest disclosure. If you want to know all about my financial conflicts, you can read about them there. But what I found is it's more useful to say these at the start of the presentation than at the end of the presentation. As you heard, I've been involved both in my role at the University of Nebraska and independently in launching a number of businesses. 3 of those are sort of going concerns right now. Ingenious Ag, which I'll mention a little bit later in the talk, was just acquired by CropX. I also advise a number of companies, and yeah, in the last 3 years I was a paid employee of Google. But let's talk about science. I'm going to tell you 3 stories today, all focused on sorghum, one about breeding, one about genetics, and one about nitrogen. This map is probably not news to anyone in this room. We grow a lot of sorghum in Nebraska, but we grow less sorghum than in many of the neighboring states. There are a lot of reasons for that, but if you talk to farmers and you ask what would it take for you to grow more sorghum, there are really 3 things. If the sorghum was more productive, if it cost less to grow, and if there was more demand and higher prices. These are all very reasonable things. And these are all things that in my program, and you'll hear from some of my colleagues at the University of Nebraska, we're working to try and address those goals. Now, plant breeding can really address the first 2 of those, and you'll hear in the afternoon, I think, about how some of the work in Nebraska Food for Health could actually address the final one, demand. But ultimately, the hybrids that folks are planting throughout Nebraska are developed and sold by private companies, and we can track how much investment, how much innovation is happening in the sorghum breeding world by looking at IP filings. This is work actually from Ravi Moral, who is a former member of my lab, now a faculty member up at South Dakota State. And we can look at how many new patented parents for sorghum hybrids are showing up from year to year. And we've got a split here because the stuff over there on, let's see, my right, your left, that's off patent now. Now anyone can use that. The green stuff is still under patent. But you can see that we had a big spike around 2010, 2014, and since then we are seeing less and less filing of new patented inbred parents for sorghum hybrids from the major seed companies. Yeah, this is usable for breeding and research. The blue-green stuff is not yet. And if we look at where those varieties are coming from, the vast majority of the patented varieties, there's a break in that graph, everything else is below 4, and then we're up around 60, is from Pioneer, which is now Corteva. So there is not the level of investment in new hybrids that we would like to see. Now, this problem is not unique to sorghum, particularly if we look here in Nebraska. It's getting harder and harder. Plant breeding is an expensive process. We have made extraordinary gains in the productivity of crops like sorghum, but every new bushel per acre is harder than the bushel before. It costs more to achieve that bushel. What you see if you look at this map, there's a very famous graph. This is in corn, not in sorghum, but it shows that yields in the U.S. have been going up about 2 bushels per acre per year, and they've been doing that pretty consistently for more than half a century. In order to achieve that, the seed companies are investing more and more money every year, and that is not sustainable. When I started as a professor at University of Nebraska, there were probably 6 companies that were really doing cutting-edge breeding effort. There are now about 2.5 companies, and sorghum, probably less than that. One of the things you do if you have to focus more and more on breeding is you look at where are the regions where we're making less money, and you stop investing in those. So what you see here is all through the sorghum belt, the yield trend, and this is for corn, not for sorghum, but we're seeing similar things in sorghum, has been essentially flat for about 2 decades, while the breeding has focused farther east in areas where the yield potential is higher. So as there are fewer companies, there's less competition, there's less investment in the region that I'm responsible for, that the University of Nebraska is responsible for, that many of you work and grow crops in. And that's a problem. So how do we lower the barriers so that more companies are involved in breeding sorghum so that there are new, better, higher yielding, more stress tolerant, and potentially higher value sorghum varieties available in Nebraska? The first thing is making sure people have access to germplasm, to starting varieties that they could use to breed new sorghum varieties. I showed you on that slide that I mean, a lot of stuff is certainly on patent. There's some stuff that's off patent. Now, as a plant breeder, one of the things that I really want is to have lots and lots of options. I want lots of genetic diversity so I can select really, really good lines. But genetic diversity is not always a good thing, and it can be a very discouraging thing when you start out. So this is a story from one of my conflicts of interest. This is Dryland Genetics, a company that breeds proso millet, a crop that has an even smaller market and an even more challenging demand problem than sorghum. We started this company about 10 years ago. We brought in lots and lots of material from across the globe, made crosses, put it out in the field, and it looked like this. It looked like weeds. It's so discouraging. We had maybe 10,000 varieties. This was the first time we'd ever gone to the field. We were walking through it. Frankly, I wanted to give up at that point. But there were maybe 5–10% of what we planted out there actually looked like a crop. And I could look over, because there's also a proso millet breeder at the university, and his field was next to mine. He had this wonderful uniform stand. All of his varieties looked good. All of them were standing. Like I said, maybe 10% of ours even looked like a crop. But we selected from those, went back through, came back next year, the year after, the year after. We are now achieving between 10 and 40% yield increases relative to what the state of the art was when we started breeding. Because we started with this really, really diverse material, we had more genetics that we could work with. But it took 10 years to get there. That's typically not the case. Okay, yeah, and here's our wonderful, this is one of the new varieties growing on many, many acres out in the Nebraska Panhandle. It's doing great. If you're starting a company, if you want to start releasing new sorghum hybrids, you probably will not have a 10-year runway. That is particularly true today. If you look at the investment climate, if you look at interest rates, there is no way to get that level of investment. So we need to move how far would a private company actually have to go. And we're doing that in sorghum. So sorghum is one of my favorite crops to work with because it is a very charismatic crop. I can take somebody who's completely uninvolved in agriculture, and I can take them to one of these research fields where we have all these varieties from around the globe. Most of these are not things you'd ever want to see in your field, but they can see this, they can immediately grasp, okay, there's something really interesting going on here. Now, ultimately, what we want is a field that looks like this. This is going to make a lot of money, that's going to make no money, but we start with that to get this and to get something that is more exciting. Again, this is collaborative with my former trainee, Professor Moral. From India, by the way, and it's really interesting hearing from him the perception of what it would take to get more agricultural exports into the Indian market. So what we are doing with this material is we have elite material, which is typically the stuff that has come off patent from the major seed companies, so the parents of modern, or at least in the last 5 or 10 year modern sorghum hybrids, and we want to integrate those with, we want to cross those with this material that I showed you in the really, really diverse slide that's been collected from around the globe, and do those first 4 or 5 years of just bringing in more genetic diversity, throwing out all the stuff that does not even look like a crop when you grow it in Nebraska, and get to that starting line where, when we think about the private sector, there may be 3 or 4 years from when they start to when they might have something they could sell, instead of the 7 to 10 that it takes for corn, or the 14 it takes often to bring a new wheat variety to market. And that makes it a lot easier to think about having more competition, having new players in this market. The other challenge we face when we bring this material in from all around the globe is most of it, the maturity is completely wrong for Nebraska. And maturity is the single biggest thing. If you don't get that right, it doesn't matter what your genetics are, you're not going to have a good crop. If you mature too early, your grain yield's too low, you've missed a chance to capture a lot more light, a lot more energy, have a lot more carbon that's going to go into your grain. You flower too late, your frost is going to kill you before you even have your grain mature. So you got to get that sweet spot right before you can even measure any of the other interesting genetics that might be useful from a production standpoint. And I'm not going to go into this part in too much depth, but this is work from a current graduate student in my lab, Ms. Harshita Mangal. And so she's using data that we're collecting from this really large population to identify really small sets of genes, in this case only about 5, that have a really large effect on the maturity time of sorghum, so that we can dial in the correct maturity and then be able to look at all of the stuff we really care about: grain yield, nitrogen use efficiency, water use efficiency, stress tolerance, lodging resistance, all of those things. And so that's one barrier to starting. If you wanted to start a sorghum company today, if you wanted to have something new, is having access to the germplasm. But there's another barrier, and it's part of the reason that we're seeing so many breeding stations close all through Nebraska, South Dakota, Kansas, which is we don't have enough people like this guy. This is Santos Rajput. He was the first breeder we hired in dryland genetics. We don't have enough trained plant breeders in the U.S. anymore. We don't train them anymore. And when we do train them, they come out of the programs where universities still have plant breeding programs, which are primarily soybeans and wheat. Those are self-pollinated crops. So we take these folks, we train them in self-pollinated crops, and then they have to go to the seed companies, and maybe they will make that jump from self-pollinated to cross-pollinated. Cross-pollinated crops would be things like corn and sorghum and sunflower for whatever reason. But there aren't enough of them. And it's really hard to figure out who a talented plant breeder is, because first they go to school, they get a degree, then they go to get a Ph.D. They're probably 9 years of education all told before they get to the seed company. And some of them have what we call a breeder's eye, and some of them don't. A breeder's eye is the ability to walk through a field like this and say, this variety, it has potential. We should really be testing it in lots of locations, get lots of data. In 5 years, it might be on farmers' fields. This one right next to it, it doesn't. And we still haven't figured out a way to find who are the good breeders and who are the not good breeders, other than putting them in a breeding station, giving them sort of 5 to 7 years after they're trained. And I saw this happen to a lot of my colleagues. You get hired, you got this job, you think you're in good shape, and about 5 years in, either you have a job or they look at your progress and they say, no, actually we don't think you're a skilled breeder, and you go away. So it takes about 15 years to get the plant breeders who can then get new varieties to market in 7 to 10 years. And as I said, we don't have enough of them. Now, if you talk to anyone in the industry, they know that this is a phenomenon. I have not found anyone who studied it in the US, but you can go to other parts of the world and it is being studied. So this is a study actually out of Ethiopia looking at wheat and teff. And when you make a new variety of a crop, initially you have only one seed of that new variety, and the next generation you have a few hundred seeds. You're not growing strip plots, you're not growing replicated yield plots. You're growing one little patch of the crop. And you can do a couple of things with that one little patch. One is you can actually harvest it. You can look at the grain yield from that little, maybe 70 square foot area. Another is that you can have farmers or breeders, and you can have them come through and just say, which of these look good to you? Does this, what do you think of this hybrid or this inbred? What do you think of this one? And you can ask them. And they'll say they like some, and they'll say they don't like others. And usually they will not even be able to tell you what it is that makes them like that one versus that one. Sometimes they do. I learned a really interesting thing about this with one of my crops, that people really liked thicker stems, because if you have thicker stems, after you harvest your crop, you're going to catch more of that blowing snow in the winter, and that melts on your field. You get a little more soil moisture. Your neighbor doesn't get that soil moisture. So you learn things just from talking to the farmers, but also the farmers, the breeders, they've seen just so many varieties over many decades. There's a lot of subconscious stuff in the back of our head that's integrating that and saying, this is a good variety, this is a bad one. So you look at the farmer scores, and then you look at the quantitative data. So we've actually measured the grain yield from this little small plot. The farmer scores are much better predictors than the actual measured grain yield of which varieties will do well in better years, in later years. The grain yield, it's noisy if you have a plot that small. I mean, maybe this plot was where the haystack was last year and the next plot wasn't. That's going to throw things off. Maybe a raccoon got into the field and, ate 3 heads. And when you're thinking about only 50, 60 plants, that's really going to throw your yields off. The quantitative data doesn't know about the raccoon. The farmer doesn't necessarily score the raccoon damage. Although I've also had the experience of growing experimental plots and finding out raccoons really like certain genetics. So we have certain plots, we lose every single head in the plot. I don't know what to do with that information, but it's information I have now. So we know this works really well. We know we can't replicate it with quantitative data, and we know it's really, really hard to train enough plant breeders. So this is work from another, actually now former PhD student in my lab, who tried to train a computer to think like a plant breeder instead of simply running a quantitative analysis of yield. So we went out, we had many, many measurements of, in this case, about 800 varieties. And we either used what most seed companies use today, which is what's called a genomic prediction model. We measure a bunch of genetic markers, we feed that into a statistical model, and we try and predict the grain yield and see how well we can do relative to the observed yield. If we can make good predictions, we know, okay, we can use this to figure out which varieties it makes sense to go on to in future years. Or what we call phenotypic prediction, which is, again, having the computer make predictions, but we're not looking at genetic markers. We're looking at basically everything we can measure about the plant. How many leaves does it have? How tall is it? How wide is the stem? What is the structure of the panicle? And it turns out this phenotypic prediction can do about as well as the much more expensive genetic prediction when we're looking in a single environment, and it actually does a bit better when we start testing across environments. So which varieties are the very best ones in Lincoln, Nebraska is going to be very different from in Grand Island. It's going to be very different from if we get out someplace like Sydney. So we can make predictions that will work across environments just from looking from these measured traits. And the fun thing about this, with genetic markers, I can go back and I can say these markers matter, these don't. That's not an interesting story. With the traits, I can get the same thing and I can actually understand something about what's going on. So we looked at this model, and wouldn't you know it, the most important thing, again, maturity, flowering time. So I told you that at the beginning, we now know something we already knew. But I can also look into many of these other traits and start to figure out what is it that the computer and probably those skilled plant breeders are looking at when they say this variety is going to make sense and this one does not. All right, how are we doing on time? The challenge, so all of these are hand-measured traits, right? This is me and my students walking through a field with rulers and notepads. And that, again, there's a limited number of people who are willing to walk through a Nebraska field in mid-July. It's 95, 100 degrees out. The steam's rising out of the mud. That's not scalable. We'd really like it if we could have computers or robotics or something make those measurements. And the way you do that is quite, I mean, you figure out if that's working is, okay, you get the computer, you get the image, you train some model to measure something from the images and say, okay, how close does that come to what a human would have measured? I am here to tell you, humans are really bad at measuring crop plants. So this is one of the traits my lab works on. It's phyllotaxy. So, you know, a sorghum plant, it should have a leaf coming out either side of the plant, opposite, perfectly opposite, like this plant on the top. In reality, some varieties do this really well. Some have a bit more trouble with it. This matters a lot when you start thinking about the architecture of the canopy. Are you going to start shading out weeds? How well will you capture light? It actually even influences water use efficiency. So we'd like to understand this trait. It's a huge pain to measure in the field if you think about going on the field like a protractor or something. So we wanted to have a computer replace that. We put, you know, okay, here are the human measurements, here are the measurements from the computer. You can see there's sort of a trend, sort of the same thing. It's kind of noisy, not perfect. But the next thing we did was say, okay, if I take two people, we're going to walk over, see the exact same plants. One person measures after the other person. The two people barely agree more than the computer. So what we can take away from this is that when the computer and the human disagree in terms of what are the properties of this plant, sorghum plant we're looking at, often it's that the human's making a mistake. So we can get better information from the computer than the human. It's not just matching human performance, it's going beyond human performance. And okay, you can say that, I mean, that's a really complicated, you know, I'm out there with a protractor and some string and trying to figure out angles of leaves. I mean, that's a hard thing for anyone to do. This one's seed color. Seed color should not be a hard trait. This is work from Nikki Shritha, another Ph.D. student in my lab, and I want to emphasize she's not the one who collected the data, she's the one who analyzed the data. What you see here are we grew this replicated field, lots of sorghum varieties from around the world. We harvested the seed, we measured the grain yield. We also had people look at the packets of seed and say, what color is the seed? And they had 8 options here. You can see folks didn't always agree. And, you know, okay, white and gray, I can see that's kind of a judgment call. Yellow and black, this starts to worry me a little bit. So, some of that's judgment call, some of that, potentially data entry errors, some of that is, we hired undergraduates for this. They would come in and after classes, you'd come in sometimes, they're listening to headphones and watching TV while they're doing the data collection. But the data, it was not so good. So we could also train a computer to look at scans of the same images. And when we have the computer collect the data, one, it's more similar between the replicates, but two, we can do a much better job of identifying the genes that control seed color in the grain of sorghum. And that's important for a couple of reasons. As you can see right over here, obviously very different market classes of sorghum. If you're going in the human market, probably you want a white sorghum, but maybe you want a black sorghum. Maybe you want something with a lot of specialized secondary metabolites that are going to enable health claims, and now suddenly you're growing a specialty crop. You're able to charge a much higher price per bushel than commodity grain. So either way, it's good to find these genes, and we find them better with a computer than with a human being. So that was a story about breeding, a story about genetics. That's my background, but I am in the Department of Agronomy. We've got to worry about production too. Nitrogen is expensive stuff. And if you look at things like 45Z, which I don't know if 45Z is still going to be a thing in 3 months. Maybe it won't be. But we are seeing a shift towards this prioritization of what were at the time called carbon intensity scores. If I talk to a different administration, I would talk about energy independence and the energy that goes into nitrogen production. Either way, reducing nitrogen applications is one of the few win-win-wins. If you can make more grain with less nitrogen, one, you're spending less money on inputs. At current prices, it's maybe $50 an acre for hybrid sorghum, probably. Maybe it's more, maybe it's less. I remember a couple years ago, we were looking at $1 per pound of nitrogen, and that price would be even higher. So being able to grow the same crop with less nitrogen is a win for growers, it's a win for the environment, and potentially it's a win for demand if we look at things like these potential ethanol credits where you will get paid more if you can demonstrate your crop is a lower carbon intensity. When I have talked to people, there's really no way this can be done tracking on an individual farm. We heard earlier, growers are already really busy. They do not need more checks and more analyses, but if we can make the case at a state level that we're using less nitrogen, that things like the GREET model are overestimating the carbon intensity score, that starts to open up more potential demand for sorghum from the biofuel sector, which is becoming a larger and larger consumer of sorghum in the U.S. All right, so that's animations didn't work perfectly. Getting to the optimal nitrogen application is in principle very easy. As you apply nitrogen, your yield goes up. At some point, it's costing you more to add nitrogen than you're making in terms of extra grain, and you stop. This can vary depending on the price of nitrogen, the price of sorghum. Should be really easy, right? And that's sort of the ideal case. Here's some data from my lab. Low nitrogen, medium nitrogen, high nitrogen, yield goes up. The challenge is that every single field and every single year, that curve is going to look different. Sometimes it's going to go up a lot, sometimes it's going to be flat regardless, sometimes you're not nitrogen limited at all. You're limited by something else in your environment, and every dollar you spend on nitrogen, every hour you spend applying it, ends up being wasted effort. But we don't know which fields those are from one year to the next. And it's an awful lot more expensive to apply too little nitrogen and miss out on that yield than it is to apply a little bit too much. The more and better data we can get you about what these curves look like in each field, the lower the nitrogen applications rates are going to be, the lower costs of production are going to be, and potentially the better the case we can make for lower CI scores for sorghum and higher value for that sorghum going into the biofuel process. Current ways of trying to estimate this are kind of a pain. So you can do soil nitrate tests pre-plant, stalk nitrate tests at the end of season, and it's better than nothing, but it's difficult to interpret. The optimal levels are going to be different depending on soil types, hybrids, weather conditions, all sorts of things like that. Also, particularly doing pre-plant applications, you're shipping this off to a facility and you're waiting quite a while to get the results back, and maybe you miss the window to get that nitrogen down on the field. So this is work from corn, and this touches on one of my conflicts of interest, but I worked with one of my colleagues at Iowa State University, And they were testing the amount of nitrate in the sap of corn and sorghum plants, and found that we can do a much, much better job of figuring out which fields will and won't be nitrogen limited from these stalk nitrate, sap nitrate tests. So below a critical threshold around 400 units, ppms, if you apply more nitrogen, you're probably going to get more grain from that nitrogen. If your stalks from sort of a V5, V7 seedling already have that much nitrogen, probably your field is not nitrogen limited anymore. It may be limited by something else. It doesn't mean you're going to have a great year, but more nitrogen probably isn't going to help. So we worked, oops, what do we have here? All right. There's a video, we're not going to try and play it today. We worked with one of our colleagues in engineering and actually developed a field portable sensor you can stick into the stalk of a plant and get a readout in the field of the amount of nitrate in the stalk of that plant. So you're getting an instantaneous readout. You're getting something that is more directly applicable to whether or not the crop is nitrogen limited. And yeah, we think this is an example where university research can collaborate with engineering, develop a product, and potentially solve a problem just by giving growers more information, not by creating more paperwork, more programs, more things like that. So we developed prototypes of this, started scaling up manufacturing, had large-scale trials in 2023, and then last year we got acquired. So this is now with CropX, and I want you to see this as an example of really, the sort of work we are doing at the university trying to address grower needs. We started out with the research in the lab, we created a startup, proved out the technology, now it's transitioned to a larger company where it can be deployed and be something that is accessible to growers. Now, I'm guessing most of you are not going to be running around your field stabbing plants in every acre. So this is a nice thing, this is not a full solution. I want to talk in my last couple of minutes about one other solution that I see, and this is something we can actually steal from Iowa. So in Iowa, what they have recognized is that they have the same problem. If we can figure out the right levels of nitrogen more effectively, we can make growing more profitable for farmers. They have a large amount of support from the state government to launch what they call the Iowa Nitrogen Initiative, where they are working with growers who have precision application equipment to run trials on every farm that gets signed up to figure out what is that optimal point on the curve for that field in that year. What is the amount of nitrogen that would have maximized grower profits? We include these usually as controls in like any experiment that we do, anything that has to do with nitrogen that Nebraska On-Farm Research does. They also do these tests where we just apply many different levels of nitrogen and figure out at the end of the season what would have been the right number. And that right number surprises people a lot. So many of you in the room already have the equipment to sign up to be able to do this, and just find out at the end of the season, after you've already got your yield, after you already put down the nitrogen harvested the crop, what was that right number? And again, we think this is a way to give more information to growers, and the more information you have, the better you can optimize, the better you can make sure you're making the most money possible. So they're up to about 270 farms all across the state. All the growers do is they get a prescription that goes into their applicator, and then at the end of the season, when they get their yield monitor data, they share that and get back this report on what was the optimal nitrogen level. I've been working with Rachel Whitehair over at Nebraska Corn, and then Adam Lease, who runs the on-farm research here in Nebraska. We're going to do a pilot this year in Nebraska. They're doing 270 locations. We're probably going to be at 10 this year. I'm hoping we'll be at more later. But I hope that if this is something that is interesting, you'll be people we can come to as we want to start collecting more of this data and both improving nitrogen use, but also making the case that when you look at these national models that are being thrown about the environmental impact of agriculture, Nebraska farmers are already a lot more efficient than I think those models account for, and we need the data to show that. So with that, thank you very much. I summarized a lot of different projects really quickly, so if you've got questions, come and find me. And I also want to thank the outstanding folks I work with on my team at the University of Nebraska. Thank you very much.